with the central dogma, we're left, of course, with this kind of problem, and that is which came first in the evolution of life. Did the proteins come first and then the coding system for the proteins come next, or did the coding system come first and then the proteins came last? Kind of a chicken or egg thing, if you will. And so much consternation has been spent worrying about this. We'll get into more detail when we talk about the origin of life a little bit later on. But RNA seems to be a good bet for the way life may have started out on Earth. We don't know the answer to this yet, but there's a great deal of work being done on it. The reason that RNA is the best candidate, and not DNA and not proteins, is because RNA can do all the things that the others can. For example, RNA catalyzes, it has enzymes which help the reaction to make the peptide bonds and the proteins. It also has a code. RNA, as you know, has a code very similar to DNA, and RNA can transfer the amino acids that are required to the messenger RNA. So RNA basically can almost do it all, if you will. So people have focused on RNA as the thing that evolved first. We don't know that, but there's a lot of suspicion out there that RNA may have been the first thing that evolved. We got a long way to go, I think, in research, as you'll find out when we get into the origin of life. But people are starting to focus on RNA as the source. Well, the important thing I would like to tell you right now is that this thing called life has the central dogma in every single bit of life on the planet, from the smallest bacteria to the largest mammals out there, the blue whale. All of them have this central dogma, or it's DNA to RNA to protein. So whatever happened in the early Earth, whatever was going on out there, we all evolved from something that created DNA, RNA, and proteins, because all life is based on that. Now, I'm not saying that when life first evolved there couldn't have been other ways to form life, but the one that won out, the one that has made all life on the planet, is this central dogma, and that'll become important when we talk about the evolution of life. I also want to bring to your attention something that's, I think, important, and that is that we have to keep this in mind, and that is that life could have come from outer space. Now, I'm not talking about aliens or anything like that. I'm talking about we find complex carbon compounds out in space, and the possibility that RNA may have formed in space, or DNA, or this whole coding system, and came to Earth and found a nice place for it to evolve and begin to evolve, is a possibility. So although most scientists believe that life evolved here on the planet, there's always the possibility that RNA or DNA could have evolved someplace else and was transported to Earth. And again, I'm not necessarily talking about aliens sending out life forms and or anything like that. It's just something that I need to mention as a possibility. All right, now I'd like to begin talking about gene cloning. And I think probably the first thing that's going to come to your mind when I mention gene cloning is you're going to be thinking, oh my gosh, cloning, that's kind of like Dolly, where you make a sheep from DNA and put it in a fertilized cell. And that's not the kind of cloning that I'm talking about. The kind of cloning I'm talking about is back in the 1960s, we didn't have any way to purify genes. And so we came up with this process called molecular cloning. It's a process by which we purify genes so that we can figure out what the genes do, which proteins they code for. So we started out in the 1960s understanding bases, but we had to and which amino acids, but then we had to find out what the genes code for, which is much more complex. And before you could find out what the genes coded for, you had to understand or you had to get them isolated and purified. So when I mention molecular cloning, I basically mean purifying the gene. And I think it's worth the time to show you this really complex method of purifying genes and how important and brilliant it was that we were able to find out the secrets of the genetic coding process through this system of molecular cloning. Again, I hesitated to put this in, but I think it's important enough to where we'll go over some of the basics. It might not all be clear because it's much more complicated than I'm letting on, but I want to give you the basics. And I want to start out with something called plasmids. Now, plasmids are these little circular, you can see them in the diagram, they're these little circular structures, and they exist in bacteria, and they have their own coding system, DNA, and we call that DNA, it's a circular DNA called a vector. And these plasmids, we'll talk about why they form, but we can separate these plasmids from the bacterium, and then we can work with that DNA vector that I just discussed. And we can also take DNA from a source that we want to find out what that DNA does. So that's what number 2 is over here. It's taking a DNA from some source of interest, and now we're going to try to get that DNA inside the vector, and I'll explain why in just a few minutes. Now, bacteria have enzymes in them. They're called restriction enzymes, and these restriction enzymes are, their sole purpose is to go out and find DNA and cut it at a very specific place. It's within about 6 to 8 bases long where they cut it. And restriction enzymes, there are many different types of restriction enzymes, so we can cut DNA at very, very specific places. So what we can do then is we can put a restriction enzyme into this system, and it'll cut that vector DNA precisely where we want to cut it. That is, cut it at the site of interest. It cuts the plasma vector, if you will, and it opens it up so that we can get the DNA of interest into the vector. Now, it'll become apparent why we want that DNA in the vector in just a minute. But I want to mention, and why would we have these restriction enzymes? Well, these restriction enzymes are part of, or are the result of, this war going on between cells, bacteria, and viruses. As you probably know, viruses inject RNA or DNA into a bacteria, and these restriction enzymes then go out as a defense mechanism, and they cut the RNA or DNA that the virus puts into the cell as a way of fighting it off. Now, this has been going on for billions of years. This is a war that's been going on between viruses and bacteria, and so there are all kinds of defense mechanisms and offensive plays, if you will, that these systems have developed. And so we're simply using what has evolved over time, and we're using it to figure out ways of understanding genes. So you can now go today and you can get a catalog from a bio laboratory, and you can actually buy restriction enzymes. There are thousands of them, and they can cut any place on the DNA that you want to cut. They've been developed for that process. So you need a restriction enzyme that cuts, between these bases on the DNA. Well, you just go and you buy it. So we can put these restriction enzymes in. They're going to cut the DNA where we want it to, this vector DNA, and then if we're lucky, the DNA of interest will go into that vector. Now, I suppose the question is coming to your mind, and that is, why doesn't the restriction enzyme cut the DNA in the plasmid? Well, it turns out that we have a methyl group, which is a CH3, which attaches to the DNA in the plasmid. and it basically tells a restriction enzyme, hey, don't cut me, I've got a methyl group on. And this is called methylase group. And we'll see when we talk about epigenetics how important this is. It becomes very important in epigenetics, which I'll discuss a little bit later on. But these methyl groups keep the good guys, the DNA that we don't want to cut, from being cut. Now, when the DNA is cut, you get a sticky end showing here, and those sticky ends, then the DNA of interest will go in, and it will go to those sticky ends and become part of the vector. Now, it won't always do that. Some of the vectors will not have the DNA of interest, but if we mix all this together with the restriction enzymes, the DNA will be cut, and the DNA of interest will go into the vector. It'll form hydrogen bonds, and then we need to take the next step, which we need to, once the genes of interest have gotten into to the plasma, then we have a new section of DNA in that vector, and then we need to seal it up. And that process I'll explain in just a minute, but I want to make sure that you understand what's going on here and why we have plasmids, because I haven't talked about that. Plasmids are also the result of a war between bacteria and penicillin. And these plasmids are out there in the bacteria, and they exist because they create proteins that attack antibodies. So this war between antibiotics and bacteria has been going on for billions of years also. And one of the defense mechanisms that bacteria have are these plasmids. And it's kind of cool because they create proteins that attack the antibiotics, but they also allow us to insert DNA that we want to know what's going on. And these plasmids then will allow us to insert a DNA of interest, and then we can put it back in the cell, as we'll find out in just a minute. But this is all the result of billions of years of bacteria and antibodies fighting. Now, in the we need to add this ligase, DNA ligase, and all the ligase does is this loose hydrogen bond that the new DNA has added to the sticky cells. It will actually make it a more permanent bond, and it'll become part of the structure of the vector. And it does a permanent attaching of the nucleotides together on the sugar phosphate backbone, and it changes it from the hydrogen bonds to this more regular bonding. And then we can take those plasmids with the new DNA in it, and we can mix it with bacteria. And some of the bacteria will pick up the plasmids. And now, here's the cool part. We've got a bacteria with these plasmids in it, and that bacteria then, when it reproduces, it's going to reproduce that plasmid in it with the new DNA in it. So this is how we can create various chemicals, and basically how the biotech industry works. Let's take an example, because right now we've just got some DNA that we put in a plasmid. But how are we going to recognize that DNA? Well, it's kind of cool. What we do is we cut up lots of DNA and we throw this in, and then we get lots of DNA, different types of DNA being added to these plasmids, and we get them back into bacteria, and then we take each bacteria, and you can put thousands, hundreds of thousands of these bacteria, single bacteria, on a petri dish, and then we bathe it in solutions that allow it to grow, and so you get hundreds, thousands of cells growing in these different localities. Now, how do we know what is what? Well, let's take an example, and you'll get some idea of how this whole process goes. Let's say we want to find the gene for hemoglobin in our library. That gene is called beta globin. So we can stick some beta globin in a rabbit, and a rabbit will create antibodies to that beta globin. And so those antibodies we can isolate, and we can actually put a fluorescent tag on it, and then we can wash it over these antibodies, we can wash it over this library of DNA, and then whatever is making beta globin, this antibody will attach to it. And since we have the fluorescent tag on it, we can recognize it immediately. And we know then that we've got the gene beta globin that makes hemoglobin. So we've basically purified the gene beta globin. And here's the cool part. When I said this was what the biotech industry does, well, that's exactly what they do. They isolate these genes, and then the cool part is they can go out and they can make beta globin till the cows come home. This has been done with insulin. So the biotech industry just figures out how to make these chemicals, and this has revolutionized medicine because of how quickly and cheaply we can make all of these chemicals from this very process of purifying genes or purifying sections of DNA that produce different kinds of chemicals. So this is the secret of the biotech industry. Now, I would like to spend a little time talking about how we began to recognize where genes were on the chromosome. This is the next step. You purify the genes, you figure out what they do, then you also have to figure out where they are on the chromosome. And I want to start out by saying that it's not too much different than what Sturtevant did in the laboratory back in the early 1900s. Remember, he was taking fruit flies and he was doing crossovers to find out where the target traits were that he was interested in. And we really do a very similar thing today, or at least back in the 1980s when we were trying to figure out where these genes were on the chromosome. So let's take an example, a very specific place on the chromosome. Somebody might have A as a base, and then every once in a while that A will be replaced with a C in other individuals. That's called a polymorphism, and it can be a genetic marker. So this particular genetic marker may be at a specific site on the chromosome, and so we can use that specific site where we see an A being replaced by a G, or excuse me, a C, that becomes a potential genetic marker. Now, and I have to keep saying this because I don't want you to think it's as easy as it appears. I've had to really kind of be careful at how much I cover here, because it would take us hours and hours to go through the process that they actually went through to make this. So I've really simplified it because of time limitations. But let's talk about the cystic fibrosis gene. We want to find out if one of these polymorphisms correlates with the cystic fibrosis gene, then we know that polymorphism and the cystic fibrosis gene are on the same chromosome. And then if we find another one that's on the same chromosome, we can do crossovers similar to the crossover studies, not exactly the same, where we can use families that have the cystic fibrosis gene, and we can also use these genetic markers in that family to determine which genetic markers are related to cystic fibrosis, and then we can see how far away they are. You don't have to do the crossover because they're already in the family. All we have to do is look at the genes and see how they correlate. So that's what this diagram shows here. This is a genetic recombinant map. It was these maps started being put together in the 1980s, and it took an immense amount of time. The first ones for cystic fibrosis and Huntington's disease, it took years to put this. Well, let me give you some data here. It took 5 years and approximately 100 scientists and tens of millions of dollars to find out where the cystic fibrosis gene is. And the way they did that was finding out how far away the cystic fibrosis gene was away from these various genetic markers. And you can see we have both male and female there. Males have less genetic markers than females. I won't go into why. but there are various percentages, there are numbers that you can see to the right of the male and the female and the sex average, and that's how far away these different genetic markers are from one another. So these are genetically recombinant maps, and this was the way that they isolated the cystic fibrosis gene. But then, in the 1980s, they said, look, this is just going to take too long if we go through trying to find all these genetic markers. So what they did is they got the whole world to combine and put all of their genetic markers that they found in various families when they were hunting for genes into this repository database, so that everyone knew where the genetic markers were, and that's what this genetic recombinant map was for. It was set up so that people on chromosome 16 could find genetic markers and then in families, and then use that to narrow down with correlations, that the same kinds of stuff that Sturdivant did in his crossovers, to narrow down where a specific gene was on that chromosome, if there was a correlation. The first human genetic map only had 403 markers. It linked almost all of the chromosomes, but just barely. And it showed the percentages of how far away these genetic markers were. But by 1995, they had about 5,000 markers. And this is the beginning then of the Human Genome Project. The project was to get these genetic markers, get as many as we can, and then make it easier to define where these genes are on the chromosome. Once again, I'm simplifying the whole process, but I'm trying to give you some idea of how the human genome, the discovery of the human genome worked. Now, if you have, let's go to this next diagram, if you have a library of large molecular clone fragments, we're talking about on the order of hundreds of thousands of genes, then you can presume some overlap between each one of those segments. And so we can cut these large fragments up and then measure the fragments. Now, I don't want to get into gel electrophoresis in any detail, but you've probably all seen this in the movies where the guys are dropping something into this gel and you see the markers on it. Well, that's a way that you can decide how big the molecule is that you dropped in there. There's a positive charge on the bottom of this gel. There's a current that goes through it. There's a negative charge at the top, and the negatively charged DNA will move towards the positive charge on the bottom. And the smaller the fragments, the faster they will move. So you've got all these different fragments, and then on the right side of the gel, you've got known weights of different molecular material. And so where all of those things end up, you can actually measure the size and the weight of the particular genetic fragment that you have. And so we can then, using this process, we can create algorithms. After you break all this up and measure it, you can create algorithms that will actually see how much overlap there must be between these, and it will then calculate how they all fit together, which is what I'm trying to show in this diagram by the molecular overlap. It's kind of called chromosome walking, and you can then use the genetic markers to find out where all of these pieces fit together on the actual chromosome. And so by April 25th of 2003, they officially declared that the human genome had been sequenced. They chose that date because Watson and Cricket published their paper in 1953, 50 years earlier, and so the human genome was officially solved on April 25th, 2003. And then after that, once they got the human genome generally worked out, the price, by the way, during this whole time too, the technology just was revolutionized. So we could analyze, you know, so fast, and I can't go into all of the details of all of the technology, the advancements during that period of time. But gosh, today, you know, what used to cost millions of dollars, now you can get your whole genome read for what, about $1,000. And now you can see where you have these polymorphisms in your own gene. You can also find out well, if you have any genetic traits for serious genetic diseases. I might mention now that we also have, by the end of the Human Genome Project, we had located more than 20 million polymorphisms in humans. And the new techniques have allowed us to map polymorphisms in families, and we've mapped over 3,500 genetically related diseases. It's quite an astonishing thing, and I want to just give you some idea of the complexity here. This is chromosome 4, and human chromosome 4, and the various genes are listed here as letters. And if you go down, I have a little red box there. That red box shows you that there's a lot of concentration of genes in that one spot. But then the blue box, that shows what they call a genetic desert. There aren't as many genes within that blue box. So the amount of genes vary along the chromosome. And it turns out that the protein coding sequences, that is genes that code for proteins in our human genome, are only 1.5% of the entire genome. I mentioned before that there are only 20,500 genes out there. Amazing. And that was astonishing to people when they got the human genome figured out. They couldn't believe that there were only 20,500. And remember, I talked to you about those regulatory sequences. Well, that's the secret. We have those regulatory sequences, which then require less genetic or less genes in the programming process because of the regulatory sequences. I should also mention that we have greater than 50% of our human genome that's made up of transposons. And these are just genetic material that floats around in our DNA and gets transferred. It was probably got incorporated in our DNA during the evolution of life from various bacteria and other things. And it doesn't serve any function. It's just what they've called junk DNA in our DNA. I want to point out, though, that that bacteria, they don't have much of this junk DNA, and that's because they're lean and mean, so to speak. They're out on the front lines of evolution, where if you've got, you know, junk, you might not get your genes passed on to the next generation. Whereas when you're in a living organism, a multicellular organism, you can tolerate a lot of extra genetic material that just gets copied on to the next generation. I might also add that when they're making that messenger RNA from the DNA, the messenger RNA recognizes the transposons and doesn't copy them. So messenger RNA only copies the gene and not the transposon. So there are ways for the messenger RNA to recognize these transposons. A couple other things to mention. There are sequences in the human genome that tend to match up in other animal life. It's about 6% of the human genome falls into this category. Protein coding that occurs across species, and this is the regulatory sequences that we've been talking about, primarily embryonic regulatory coding systems that I mentioned before, telling, you know, okay, you need to be a bladder cell, you need to be a skin cell, that sort of thing. And those regulatory sequences become very important later on. Just a quick note about cancer. Cancer is the mutation of a single cell where it has runaway growth. So the idea then is that we can go in and we can analyze that cell that's mutating, and we can get the genome of it, and we can compare the cancerous cell's genome to regular genome and find out what has caused that cell to mutate and have runaway growth, and then we can target the process that's causing the runaway growth and block it. So this has opened up a whole world of new genetic medical techniques that are being, as we speak, put into effect.